Thank you very much for the kind invitation to speak here at this course. So it's a great pleasure to be back in Paris. I think it's been here last time more than 2 years ago. And yeah, today I want to talk about the physics of biological condensates. That's a subject that we have been very interested in Dresden since now more than 10 years. It is the phenomenon that in cells, biochemical compartments are organized by the condensation of proteins, usually together with nucleic acids, and they form sort of droplet-like structures in the cell. Let me first also acknowledge my co-workers, because probably at the end I will not have time. So this is the collaboration of two Max Planck Institutes in Dresden, and we have also a joint center for systems biology, which is sort of a hub to integrate collaborations between theory and experiment. So this is the outline of what I want to do today. Also depending on time, I'm not sure how long I can speak now that we started late, but I guess you keep the time. So I will first sort of give an introduction and talk about what are biological condensates in cells. Yes, then I will be talking about active emulsions and active droplets. That's motivated by the question of how does droplet formation and chemistry work together? What kind of phenomena arise from that? That's sort of the main part of this lecture. And then towards the end, I will also talk a bit about the role of condensates in the organization of chromatin, and if time allows, also talk about material properties of these sort of. So cells have a complex spatial organization to organize their biochemistry. The classical picture of how this happens is to separate different biochemical compartments by membranes, and that's a sort of the picture of the classical organelles. However, there are many biological compartments or chemical compartments that are not organized by membranes, membrane-less compartments or membrane-less organelles. Here you see sort of examples both in the cytoplasm and in the nucleus. Yeah, so typically these structures combine nucleic acids and proteins. So there are many RNA protein condensates, I shouldn't use that, I guess. Condensates, and there are also DNA protein condensates. This research by looking at P granules in the C. elegans embryo, which are shown here as these green objects in the cell. And these are an example of RNA protein condensates. They contain many RNA binding proteins and RNA. They don't have a membrane, and they play an important role in the specification of the germline of the organism. Now here I wanted to show you a movie because what happens during cell division, to the asymmetric cell division, is that these granules segregate to one side of the cell, and only one of the two daughter cells will have these granules. So there is a physical separation of molecules in the cytoplasm towards only one of the two daughter cells during asymmetric cell division. And in this dynamics, one also can see that it's sort of an emulsion. The cell cytoplasm is an emulsion of many droplets that are then segregated. And from this picture of having condensed objects in the cell comes the idea that an important concept here is to think in terms of liquid demixing as a key idea in order to think about these condensates, so that you have a mixture of different molecules which can undergo a phase separation into two coexisting phases that have different composition, and this then depends on temperature or composition, which can regulate these processes and can be characterized by phase diagrams. They then also demix and form droplets. So here you see purified pregranule protein PGL3 in buffer, and this is an emulsion of droplets formed by these proteins. So in this movie, they would be falling down and then collecting in a fluid in the bottom, because they have a higher density than the buffer. And here is a picture of an emulsion, which actually is a movie in which you see many emulsion droplets ripening, as in the classical emulsion. Droplets can also fuse, showing that they are really liquid droplets. So these phenomena can be understood with classical thermodynamics of phase coexistence, so free energy, which depends on composition. The density of this free energy is of a local quantity, can have a curvature that changes sign, and as a consequence there can be a coexistence of two different states which have the same chemical potentials, same pressures, same temperatures, and this can be then identified by such a Maxwell construction. The slope of this free energy density is a chemical potential. Equality of chemical potential means the slopes must be the same, and this Maxwell construction identifies the coexisting compositions of two phases. Now use notations, concentration will be little n. Sometimes you use volume fraction, which is concentration times molecule volume, phi. And from this picture of having two coexisting compositions, then arises such a phase diagram where there's a range of composition in which the initially homogeneous system demixes in coexisting phases. Here we have a binary system. There would be a B-rich phase and an A-rich phase that coexist. Now, when we have an emulsion droplets, the equilibrium condition at the interface is modified by curvature of the interface. So the surface tension induces in the pressure balance the Laplace pressure, and it shifts the values of the chemical potential. So the chemical potential are still the same locally inside and outside the droplet. So the slopes here must be the same, but there is a shift between these two lines by 2 gamma over r, where r is the droplet size. And therefore, the local equilibrium is affected by the curvature and droplet size. And this then gives rise to this ripening phenomena, that a smaller droplet has locally higher chemical potentials than a larger droplet with the same sort of phase coexistence, and there is a diffusive flux of molecules from small to large droplets, and droplets will ripen, which would be shown in this movie if it would run. Now, the physics of droplets and liquids can also be observed in the living system. So in this C. elegans model, one can, for example, since these P granules exist in the germline, in the adult worm, one can look in the germline and finds these P granules wetting the cell nucleus. So here you see cell nuclei, which are wet by these droplets, which are fluorescently marked. And these droplets behave like liquid droplets. They can fuse in the system, and one can estimate quantities such as viscosity and surface tension just by looking at the dynamics of fusion and by looking at diffusion of particles in the cell. So here, typical values of the surface tension one can estimate here, and the ratio of viscosity and surface tension can be estimated from the dynamics of fusion. Now, in the cell, of course, equilibrium concepts do not apply in general, and it's an interesting discussion about to what degree and in which conditions can we use equilibrium thermodynamic concepts. Now, the first interesting question is whether these emulsion droplets behave as one would expect thermodynamically if one changes the temperature. And here I show such an experiment. I can't run a movie, but in this experiment, the temperature is changed from 50 degrees to 27 degrees on the upper embryo, and it's left constant at 70 degrees at the lower embryo. And what one finds then is that the droplets actually dissolve. And this is reversible. So this would happen as the temperature is increased. They're dissolved, and then one can reduce the temperature again, and the droplets will reappear. So this looks a lot like a phase separation phenomena, even though one looks at it in a thermodynamic phase separation phenomena, even though it's under non-equilibrium conditions in the cell. And one can even sort of measure something like a phase diagram. So here you see, as a function of temperature, the concentrations measured of this fluorescent component outside the droplets and inside the droplets at different temperatures. And this is sort of like a binodal of an equilibrium phase separation. And one can, of course, the system is very complex. It's a multi-component system, but it can be sort of fit by an effective two-component Flory-Huggins model, where we take into account that the coefficients in the model are sort of in general temperature-dependent functions. And this then sort of raises the question of whether this is a good picture. Under what conditions can we think of a local equilibrium phase diagram? And is temperature a well-defined quantity in cells when we control it from outside in an experiment? You see this 20 degrees and you see that 30 degrees. The first question one can ask, can one control the temperature in a cell? And to discuss that, I have here the temperature equation, which satisfies essentially a diffusion equation with sources. Now, in the cell, there are processes taking place that release heat. So when chemical reactions take place with an enthalpy change in the reaction, there will be heat generated, and temperature will be obviously the diffusion equation, and temperature, of course, can be dynamic. And we have boundary conditions so that the temperature is set from the outside. There's some noise here in the background. So in general, an organism or a cell releases heat, and the typical order of magnitude is 1,000 watt per cubic meter. It's sort of the heat production by the order of magnitude of an organism or a cell. And this gives then rise to a non-constant temperature profile. But this temperature profile changes over long distances. So the typical length scale here is of the order of tens of centimeters or meters. So in a cell, the temperature is very much homogeneous, and it relaxes quickly. So if you put the numbers in, it relaxes sort of in the order of milliseconds if an experimentalist changes the temperature outside. So we can control the temperature from outside, and it's constant in the cell. A second interesting question, and we want to discuss it on the blackboard, is now what happens if in the cell chemical reactions take place and release heat? This generates fluctuations. And should we think of these fluctuations as very strong? Can we think of local equilibrium, or is there no local equilibrium in a cell environment? And to discuss that, let me look at a sort of a simple-minded estimation. So I'm starting from the equation for the temperature dynamics. It's a diffusion equation with a source. Q is the heat generated per unit time. Rho is the mass density. Sorry, this is the C. And Cp is the specific heat, the constant pressure. And now imagine that there are these discrete events where ATP, for example, is hydrolyzed, and this releases heat of the order of 10 kBt, which is the enthalpy change of an ATP hydrolysis. And then the heat rate could then be written as sort of a sum of many of these events, Hi, which occur at a position xi locally and at time ti. And now we have a superposition of many of such events. And let's look at a single one of those. Let's say t equals 0 and x equals 0. Then we have a perturbation of temperature which has an amplitude hi over rho Cp, and then behaves like a Gaussian that spreads. So alpha is the heat, the diffusivity of temperature. It's just a spreading Gaussian, where I just use a solution to a diffusion equation with the delta source. And here one can now plug in numbers and ask how strong is the effect, and is it sort of self-consistent to use an equilibrium argument. And if we now put in very short time scales here, let's say the first 100 picoseconds, this event has spread over a distance by this diffusion sort of law. So that's the spread of this event is diffusive with a diffusion coefficient which is given by the thermal conductivity, by the diffusivity of temperature. Also, next slide. So after t equals 100 picoseconds, we would have a delta x of about 1 nanometer and a delta T of about 3 Kelvin. That's the strong perturbation. If we go to about 10 nanoseconds, that's 100 times longer, we spread over about 10 nanometers, and delta T goes down to about 3 millikelvin. And if we go to 100 nanoseconds, sorry, 100 nanometer, 1 microsecond, we have spread over 100 nanometers, and this goes down to only 3 microkelvin. So this, you can see from this perturbation decays very quickly in amplitude and spreads in a diffusive manner with time. And you can take from here the message that if you look at volume elements of the order of 100 nanometer size, and you're interested in time scales that are longer than microseconds, then the system is essentially locally equilibrated in these small volume elements. So these are big enough so that you have many molecules, and you can do sort of locally thermodynamics, and they're small enough that you are not yet sort of seeing large-scale non-equilibrium phenomena. So the logic here is that on small scales, let's say, as an example, 100 nanometer volume elements, in slow processes beyond microseconds, we can use local equilibrium arguments and then build from the local thermodynamic equilibrium many different volume elements in space on larger scales and non-equilibrium physics that emerges only at larger scales, but based still on the local arguments of equilibrium. And that's what one usually does when one applies irreversible thermodynamics to non-equilibrium processes. And in order to now describe, for example, droplets in the cell, what one essentially needs is that the droplet interface between droplet and outside is locally equilibrated, and that the jump in concentration and composition between inside and outside can be captured by such thermodynamic arguments. Then they can use sort of many of the concepts, but then of course at larger scales there can be concentration profiles, there can be diffusion fluxes, and there are non-equilibrium processes happening in the cell. And this is a very simple-minded argument. So there may be conditions where this is no longer applying. If you go to membranes, it may be more complicated to surfaces. But this gives us tools to decide whether our certain approaches are self-consistent, and we can also try to understand when we have to go beyond local equilibrium arguments. Yes?
Can I ask a question?
Sure. Yes, I should also say, please interrupt me and ask questions. That's a very good thing to do.
So we see that all these are happening so fast, in a order of picoseconds. But when I saw the movies, the image, it was like 9 minutes. So why do we need to like all these in a long time scale if all these are happening in like event?
So the fast time scales here is the dynamics of a local perturbation that is fast when you have a chemical event that is rapid. And the question, because the chemical event that is rapid is clearly driving the system somehow out of equilibrium. And the question is, under what conditions can we still use thermodynamic arguments for the slow processes that we're interested in? Yeah, we're interested in rather slow processes. Many enzymes operate on milliseconds, or if you look at longer time processes, they're even slower. If you look at these condensates or droplets, there are many slow dynamics. And because there's now a separation of timescales. Yeah? And the point is that if you look at slow processes, local volume elements have the time to equilibrate, even if at short times they are not, things happen that are not consistent with equilibrium. Does this answer the question?
Yes, thank you.
Yeah. Let me now come back to the P granule. So the idea is now you can use sort of local equilibrium concepts as interfaces between let's say, compartment condensate and outside the condensate. And we now like to understand the larger scale dynamics, yeah, which is non-equilibrium. An example is here the segregation of these P granules during asymmetric cell division to one side of the cell. And when we look at this process in the cell, what drives this segregation? The first idea was that there are flows, which you could have seen in these movies, but these flows cannot really net transport material to one side because they are circular. So when there are flows to one direction of the center, there are backflows on the sides. So what actually segregates the material is a gradient of assembly and disassembly of these condensates. So they assemble on the posterior side and disassemble on the anterior side. And this assembly-disassembly is organized by a concentration gradient of a protein in the cell. There's a protein called MAX5, which is a high concentration at the anterior and the low concentration of the posterior. And so we have a situation where in the cell there are gradients, and sort of the local equilibrium conditions are position dependent, and therefore the system is overall not at equilibrium. Already the active processes are needed to set up such a concentration gradient and to maintain it in the cell. So here you see a measurement of this MAX5 protein concentration, and here you see the segregation of the P granules. So in this situation, then we have at the high MAX5 concentrations, P granules tend to dissolve, and at low MAX5 concentration, they tend to grow. So in this case, the idea is that compositional variables regulate local phase coexistence. And the picture that has emerged in this work, which was then in collaboration with the group of Toni Heimann and also with the group of Christoph Weber, is that the coupling between MAX5 concentration and P granule thermodynamics goes via the binding of RNA. So the PGL3 phase separates in an RNA-dependent manner. RNA promotes condensation of PGL3, and MAX5 is also RNA-binding protein. So we don't need a direct interaction with MAX5 and PGL3, but by MAX5 binding to RNA, it reduces the local RNA concentration, and then this favors phase separation of PGL3. So this is a rather complex multi-component phenomenon that then leads to an effective phase diagram where the PGL3 phase separation is MAX5 concentration dependent. And this can be then sort of simplified in a diagram which is more schematic here, a very simplified model, which has PGL3 density and MAX5 density, and I don't look anymore at the RNA concentration. So it's an effectively reduced diagram. And the idea then is that we have a phase separation of PGL3, which is dependent on the local max 5 concentration. In the cell now, as the cell gets polarized with a concentration gradient, different sides of the cell will be represented by different points in the phase diagram. Of course, the phase diagram only describes local equilibrium. It doesn't describe, the cell is out of equilibrium, so it cannot be described by a phase diagram, but every point in the cell one can think of corresponding to a position on the phase diagram. And now we can have a situation where the two ends of the cell lie in the two regions of the phase diagram. The posterior side is in the two-phase region, the anterior side is in the one-phase region. Overall, the system cannot be at equilibrium, and but we get a situation where droplets grow on one side and shrink on the other side. And if we now take everything together, we have an emulsion in the concentration gradient. And that's, of course, a very non-equilibrium phenomenon. And I show you in the simulation of an effective droplet model. We start out with sorry, I can't show you the simulation because it's not a movie. So what you would see here is first an emulsion of many droplets, homogeneous, and then we introduce a gradient of the supersaturation or a gradient of the other component controlling phase separation. And then droplets shrink on one side, and you see that the material in the emulsion is shifted towards the side where droplets grow. Okay, so that's sort of the starting point as an example of how to look at different scales and how the cell can regulate and organize the condensation of such granules. Of course, more generally, these condensates are biochemical compartments which have certain composition and which carry certain biochemistry. So the idea is that this condensation organizes the chemistry of a cell, and can organize it in space by localizing chemical reactions into condensates. So in the condensates, you can collect certain enzymes and certain specific reactions that take place in the condensates. And you can think of them as sort of as little microreactors that carry out certain tasks and certain types of biochemistry. And this theme and this idea brings us to the physics of combining now chemical reactions with phase separation, and to develop the theory of chemically active emulsions and chemically active droplets. And the approach that we've used is sort of in the spirit what I outlined, that you can use locally at small volume elements thermodynamic arguments. You can define locally temperature, chemical potentials, and free energy densities, and then you can build non-equilibrium physics at larger scales from there. So the starting point is a description of the thermodynamic with a free energy density defined locally, and then in order to describe phase coexistence, but also these gradient terms that then generate the interfacial tension and the thickness of interfaces. Now this free energy in general depends on the concentrations of all the components, and we need in general a multi-component approach. And from such a free energy, one can, it has a shape as illustrated before. And if it has these changes in sign of curvature, it typically has regions of phase coexistence. And if we go away from thermodynamic equilibrium, we use what is called irreversible thermodynamics, where you allow different neighboring volume elements that are in states that are not together at equilibrium, only locally, and then changes in, for example, chemical potentials between different positions then drive diffusion fluxes. So the chemical potentials are defined as the functional derivatives of this free energy with respect to the concentration profile. And we need a functional derivative because of this gradient term. And at equilibrium, these chemical potentials are equal everywhere. And this can also happen if you have, as I mentioned before, if you have coexisting phases, if chemical potential is the same inside and outside. But as the system goes out of equilibrium, you have gradients of chemical potential, and they now drive diffusion fluxes and transport of molecules in the system. So the dynamic equations for concentrations are now governed by these diffusion fluxes. And the next step is now to add chemical reactions. So you have a emulsion of droplets that are dynamic, they may ripen, described by this thermodynamics. And now we have these components undergo chemical reactions. And this will then give rise to source and sink terms in this equation. And to be more specific, one can quite generally write down a general reaction scheme for many components. So AK would be a chemical symbol of the component K. It can be a molecule, molecular species. And sigma are the stoichiometric coefficients of the chemical reaction. Sigma plus are those on the left side of the reaction, and sigma minus are those on the right side of the reaction. And here you have to sum over K. So this is a very general way to write. Every chemical reaction can be written in this general form. And then the reaction has an index alpha. So the reactions, alpha goes, let's say, from 1 to the number of reactions. And for each reaction, we have such an equation, which takes a number of molecules on the left, which react to the certain number of molecules on the right side. Yeah? Maybe add a few things here. Sort of the idea then is that I can define a stoichiometric matrix sigma I alpha equals sigma I alpha plus minus sigma I alpha minus. That's sort of the net change of the molecular component I in the reaction alpha when it happens. And one can then define the chemical potential or the chemical free energy that changes by one reaction event, or usually one calls it a Gibbs free energy change, delta mu alpha, which is then sum over K sigma K alpha mu K. So mu K is the chemical potential of species K. These are the stoichiometric coefficients. This defines the chemical free energy change that happens during one reaction event. Now, the reactions happen at a rate Rk, sorry, R alpha. Alpha is the index for reactions. That's the number of reaction events per time and volume that takes place. And I write this as a difference between a forward rate and a backward rate. So that's expressed. So this R plus is the rate at which it runs forward, and R minus the rate at which it runs backwards. And now thermodynamics sets conditions on these chemical rates. So at chemical equilibrium, the forward and backward rates are equal, and there's no net rate. And chemical equilibrium means that delta mu alpha is 0, that there is no net chemical free energy change during a reaction event. Now, if, to be consistent with thermodynamics, there's a detailed balance conditions for the chemical reactions, which is associated with micro-reversibility, and which is written here, which is that the forward rate divided by the backward rate must be the exponential of delta mu alpha over kBt. There's a typo here because the kBt is missing. And from that, it follows that the rates can be written in a general form, which is some kinetic coefficient, which is in general composition dependent, times factors which are thermodynamic. And in the forward and backward rate, I have the same kinetic coefficient. And then the thermodynamic factor. And with this, I have now a general theory that allows me to capture chemical processes and phase separation processes that are consistent with local equilibrium and can be extended to global non-equilibrium. Yeah. Yes?
Thanks. Just a quick question. Why do you have this gamma as the diffusion coefficient? Does it introduce some kind of anisotropy in your system, or?
You're talking about the kinetic coefficient here.
Yes.
Yeah? So the gradients of chemical potential drive diffusion fluxes, and this is phenomenologically described by a coefficient in so-called linear response theory. There is a linear coefficient describing this relationship. That's what is gamma. And I simplify it here because in general this is a matrix if you have many components. I just didn't want to make the slide too messy. And this is related to diffusion coefficients, but it's not the same as the diffusion coefficient because you have first to translate, you have to put in how the chemical potential depends on concentration, and then you will see that there are second derivatives of concentration, then you can identify diffusion coefficients. So this is related to diffusion coefficients, but it's not a diffusion coefficient the way it's written. Yeah? Okay. But the matrix of gammas defines the matrix of diffusion coefficients.
Thanks.
And so the kinetic, so we have thermodynamics. The kinetic coefficients are these coefficients gamma, which can be translated to diffusion coefficients, and these coefficients k, which then determine the reaction rates. And these coefficients together then give all the information one needs, together with the thermodynamic information that we have here. So this is sort of the general framework. What is often useful is to drive these systems out of equilibrium in the sense that we use, for example, ATP as a fuel. We don't care about where it comes from and where it goes. We just assume there is a maintained concentration. And we can somehow describe the system by focusing only on the concentrations of interest and leaving out sort of the fuel as a variable. And this then amounts to adding active contributions to these reaction free energies. Yeah? So the fuel, of course, enters the chemical reaction, but if I don't want to describe it explicitly, it's enough to just add an extra chemical force to these chemical potentials, and this allows me then also to drive the system out of equilibrium by maintaining the fuel out of equilibrium, for example, having ATP at high concentrations as compared to ADP. And this is sort of a natural way to introduce this into this framework. Now, with this framework, we can now look at droplets with chemical reactions. And there are two different types of systems that are interesting to distinguish. And the first one is using droplets as containers for chemical reactions. So then in this case, the example would be an enzyme in the droplet having a substrate turned into a product. So then the droplet can localize reactions, can concentrate reactants, can maybe accelerate the reactions. That's of course relevant for biology. It's one type of system we can think of. The second type of system, or there's a type of it should be two, is a system where the droplet itself is chemically driven. So where the droplet grows because of reactions or shrinks because of reactions. So that's when droplet material itself is turned over by the reaction literally. And that's what is really the core of the concept of active droplets, because they become really very different from the classical emulsions. So we find novel behaviors. An example is a binary mixture where we turn A and B's into each other inside and outside a droplet. And so we can look at a sort of a minimal system to discuss these features. And let's first start with a two-component system. And in the simple picture, we can just describe phase coexistence between local equilibrium, and then we have inside and outside diffusion reaction equations. So then for the two components, I would have diffusion reaction equations with source terms. And these reaction rates now are linked to thermodynamics in the way I described before. Now, if we let a system run without sort of adding some sort of chemical fuel, because A can be turned into B, there's no longer the conservation of the material that defines the droplet, and therefore the system can minimize its free energy by just going to the minimum of this free energy functional. So what typically happens in this simple binary setting is that droplets dissolve if the chemical reaction is switched on. But by keeping the system outside of equilibrium, by adding sort of this idea of a chemical fuel, where the reaction between A and B is sort of driven by some other components, you can think of ATP, ADP hydrolysis, which effectively breaks this DT balance condition in the original way for these forward and backward rates, then droplets can persist. So one can sort of generate a situation of stable droplets out of equilibrium, which would not happen at equilibrium because of this chemical reaction. And the first interesting observation in such a system is that the ripening can be very different from the standard ripening, standard Oswald ripening. So these would be two movies. The left one is an emulsion that ripens without a chemical reaction term in my dynamic equation that is solved here. And the right one is with an added reaction term that has a broken detailed balance because of an extra delta mu from a fuel. And this gives to an arrested ripening. So this was the droplets which go to same sizes, and it doesn't ripen anymore. So we call this suppressed Oswald ripening. And that's a very different behavior from normal droplet ripening. Now, what this means is that such a droplet attains a specific size. So rather than growing, it goes to a certain size and stays there. If it's larger, it would shrink. If it's smaller, it would grow. And this also led us to think of these systems as very primitive models of cells. They don't have a membrane. You can think of as physical objects that are sort of similar to cells and sort of prebiotic cells, if you want to think of how the first cells emerge from simple structures. Because this somehow is a well-defined object which runs chemistry, has a certain size, it's out of equilibrium, and has some sort of a turnover which you can think of as a metabolism. And if you, one can take such a system and plot the rate of growth, so R would be the radius, as a function of the radius itself. In a normal system without chemical reactions, there's a critical radius below which you need to nucleate the droplet. If you go beyond the critical radius, it will grow, but then it will always grow. While with these chemical reactions turning it over, the curve changes and has another intersection with the zero line, and this is a stable size that arises. And now there's something interesting, that this stationary system, a stable site, can become unstable in terms of shape. So this is related to what is known from, for example, crystal growth, from the Multiscale instability. When you have an interface that grows because material flows to the interface, there can be instability where the interface starts to exhibit a wave-like perturbation, and somehow the wave grows faster at the tip than in the wells, and it's called the Multiscale instability. On the droplet, you can have a similar thing. If the droplet grows, in principle, you can have such instability as well. But what is particularly interesting in our case is because the droplet does not grow, because it's stationary, but it turns over, so there's a flux of material to the surface. You can get this type of instability at constant volume. And that means that it changes its shape by extending in one direction, but it narrows in the perpendicular direction. And as a result of that, it can undergo division. I think that was a question.
Yes. Can you go back to the slide? So the upper curve without the chemical reaction correctly predicts that. Yes. So this essentially predicts that at some point there will be one big droplet. It's a complete phase separation in
system? I mean, if it continuously grows.
Yeah, this depends on exactly on the ensemble you're looking at. If you have a large box, which has a fixed volume, and you have a certain amount of molecules, then it grows until the size that is set by the composition of the large box. And if you now take a thermodynamic limit, there are different limits you can take. But if you have a thermodynamic limit, then with the volume, also the droplet size will diverge. And so there is a thermodynamic limit where the droplet grows without bounds. Yeah. So these are snapshots from a calculation that shows how the shape becomes first unstable with an elongation, and it goes all through division. I also have a movie of that, but I can't show it. So that's the initial configuration, and it will then extend and split. And I'm solving the simple equation I showed before with a diffusive term and the reaction term, and it's out of equilibrium. And this is sort of a state diagram, which essentially, so we have an A-binary system. Inside the direction B to A dominates, outside the direction A to B dominates. There are parameters governing those two reactions. They have broken detailed balance to maintain the out of equilibrium. And sort of the two axes essentially are the parameters describing the reaction rate A to B and reaction rate B to A. And if you somehow make the reaction rate B to A very strong, then droplets tend to dissolve because they disappear. If one increases the A to B reaction, then one can have stable spherical droplets with well-defined size. And further increasing this reaction rate, one gets the shape instability and droplet division. And now when a droplet divides, after division is smaller than before. So it's below the preferred size. So the two droplets generated will grow again until they reach the size at which they would be stationary, but then they undergo the shape instability again. And so you can get cycles of growth and division in these systems. And they have also a lot in common with, they look very similar to cell divisions, even though they are much more primitive. In particular, the axes of divisions are oriented in ways that depend on where the neighbors sit. So one gets a phenomenon where first division is arbitrary, you get two new droplets, but they now divide on an axis which is perpendicular to the axis between the two droplets. And this also happens with the third division. So you get an arrangement of cell division axis or droplet division axis, which is very similar to what you actually see in much, much more complex biological systems. And another aspect now in the system, which I so far sort of not mentioned, is that this also couples to hydrodynamics, to fluid flows. Usually if you have an emulsion, droplets are spheres. And then the Laplace pressure is the same everywhere on the surface of the sphere, and there's a well-defined pressure inside, there's a well-defined pressure outside. You don't couple this usually to hydrodynamic flows. But here we generate shape changes by the chemical activity. So the chemical activity somehow works against surface tension, deforms these droplets. And by doing that, the curvature is no longer constant on the surface, which means that the Laplace pressure is now a variable along the surface. This then implies that there are pressure gradients. And these pressure gradients now drive hydrodynamic flows. So what one in principle has to solve is the system which adds to everything which I described, also the hydrodynamics. So we have now an extra convection term in our concentration equation. This is the diffusion term, this is the convection term, this is the reaction term. The flow velocity is, in the simplest case, that is described by a Stokes equation. I take incompressible conditions. I have now a pressure field, and the pressure field now can account for the Laplace pressure field, which is already governed by shape. And the simple picture I just introduced before emerges when the viscosity becomes large. Then these flows become less and less important. And here you see a state diagram taking into account flows. This is the region without droplets. Here are stable droplets, and here is the shape instability. You still have all the phenomena, but the phase boundaries change and are shifted a bit in a viscosity-dependent manner. Another aspect is to take into account fluctuations. And here I would like to show you a lattice simulation, which is of a Metropolis algorithm to generate dynamics. You take a lattice, you make moves of concentration between lattice sides. It describes a fluctuating diffusive system, and we make stochastic moves associated with reactions on a given side. And then one can use a Metropolis rule to define kinetic rules that are consistent with the thermodynamic equilibrium, but also describe the kinetics. And by coupling this now with to a delta mu from a fuel, you can use the active system. And this movie would show you that this fluctuating droplet also divides. So we can use all these tools to capture different aspects of this problem. Now, what I showed you is droplets that divide in 3 dimensions. And it turns out that the dimensionality makes a big difference if you compare this solving the same equations in 2 dimensions, which is much easier. So it's appealing to solve these problems in 2 dimensions as toy models, because it's much faster to do on a computer. It's much easier to do. 3 dimensions is rather slow. But there is a big difference. And this has to do with sort of Rayleigh instability. The Rayleigh instability of a cylindrical fluid helps dividing droplets, because it makes narrow necks sort of potentially unstable. It doesn't work anymore. So this is unstable, and this thing tends to split. And in 2 dimensions, this is not unstable if you have a little 2-dimensional connection like that. And it doesn't actually narrow. And as a result, if you solve the same equations in 2 dimensions, I personally have a movie here, but what happens is there is instability. It starts to extend, but it doesn't split, and then there's things like that. And you get one structure that grows in an unstable way, but it doesn't split in different objects. Now, there are ways to make droplets also divide in 2 dimensions. One has to add extra terms, and that's some of the sort of ongoing work. But essentially, one can use chemical reactions that are sensitive to the curvature at the interface. Somehow I need a different pointer. This doesn't work anymore. Not sure what it is, but got the old-fashioned.
Okay, that's great.
Is it back? Yeah, but it's not very stable.
Okay, I will try to have more battery.
I'm not sure it's the battery, because it's harder to use it. So by adding extra terms on the reactions, one can destabilize the necks and have droplets divided in 2 dimensions. I can't show you the movie where I start from a single cell and a single droplet. I call them cells, but they look so similar. But, and to get a whole field, you can sort of have a wave of growth and generate more and more droplets. What happens if you start with a single one like this is that they divide and you generate more and more ones, but they only divide near the boundary, not in the center, because there's not enough material in the center to feed them. But they can go in the boundaries. You get a growing region of droplets that result from division. Now, so far I've illustrated the basic ideas using a 2-component system, which is extremely simple, oversimplified. If we want to build models for, let's say, protocells droplets, which can be models for primitive prebiotic cells. We'd like to have more components. And here I show you something which I would think of a minimal model for a protocell, where I have D is the component which forms the droplet that phase separates from the solution. But D is not directly provided from outside. So there's some component N, which I call a nutrient, which is dissolved in a medium, and it diffuses in the droplet, and then the reaction inside turns it into D. And then there can be another reaction inside D, which turns D into some waste product, which can then diffuse out of the droplet. And to describe this system, I need 4 components. But
Try the black one. The one.
This one?
Yeah.
I'll try.
The pointer in the course.
Let's try. It does not.
Like that? Just the pointer?
It comes from this pointer here, but it's not doesn't really work. I can try it, but it always blackens out my slides. No, it doesn't work. The moment I point, the slide disappears.
I'm trying to get
Should I use the mouse? No, it's very slow. So I need 4 components to describe this type of system. If I describe it with the volume fractions, they all sum up to 1, so there are only 3 variables left. I could try, but the mouse doesn't really work well.
You can use this part.
Yeah. Okay, that could work better. Let's try that, yes. Now, when we have more components, we have to take conservation laws more seriously. So these reactions, of course, imply that the 4 components are not conserved. But there is still a conserved density, which is the sum of those 3 components. Because the reactions turn one into the other, and so the sum of them is not changing. And these conservation laws, then of course, means there is contradictive sign now satisfy the conservation law. And these conservation laws are key because if a droplet wants to grow, it can only grow by the conserved quantity arriving at a droplet. So we can discuss here sort of the constraints to the conservation law. So droplet growth happens via the flux J in psi. And you cannot see from the fluxes in the individual quantities whether this allows a droplet to grow or not. And of course, these are constraints which you also have if you have an organism. To grow an organism, you have to bring actual conserved quantities to the organism in terms of matter. And a similar discussion exists also with energy. So you have energy balance and you have a matter balance that has to be taken into account. And these primitive models allow you to discuss the physics of all of this. So the second point is the energy supply. So we have matter supply, we have energy supply. And energy in this case is supplied by providing, either by maintaining the chemical potentials with nutrient waste at different levels. And then you need an active process maybe to renew it. That would be my chemical fuel in the earlier pictures. But we can also just supply it at the boundaries. You can bring the system in contact with the reservoirs of nutrient waste, and you have to make sure that the reservoirs keep them at different chemical potentials, so that the nutrient really provides then the free energy. So for that reason, one can distinguish here 2 different ways to drive the system out of equilibrium. Yeah, on the left is the picture I showed you before, where I use some extra energy input, delta mu, of component C, that somehow breaks the detailed balance between W and N, the waste and the nutrient. Here the waste is sort of used to recreate some higher energy nutrient. And that's what we call it, it's driven in the bulk. So there is something happening in the bulk that puts in the energy into the system, that drives it out of equilibrium. And physically, that could be, for example, radiation like light. Yeah, if you think of how plants get energy, that would be the type of process. While the other way to do it is sort of via reservoirs, via boundaries. You provide the nutrient in an N reservoir and the waste in a W reservoir, and you have now permeable walls. But now there's no breaking of detailed balance inside the box. So all the rules are thermodynamically consistent with detailed balance, and the only breaking of detailed balance is via the boundaries.
Yes.
Yes?
So in this model, you
So for the question, look for the numbers, please.
okay. So in this model, you assume that there's already a droplet, or can it also emerge from N with no D? So do you need like a primary condensate, or it can
And now you ask about the
now
you ask about the nucleation problem. You know?
Yes.
That's of course the generalization of the question of do you need a droplet to get a second phase, or how do you nucleate a new phase if you are in an existing phase that is stable? Yeah? And that's of course a complex question. The first answer is in principle you can nucleate new droplets, but then the question, how does this happen? If in nucleation theory you usually need fluctuations, because if you have really a stable phase, without the fluctuation it will not happen in the first order phase transition, unless you go beyond the spinodal instability and then things happen, but it's also very complex. Here I usually use models without fluctuations, and then I need to start from a droplet to initiate it, and then I can get very complex phenomena. And you can think of the question of having droplets and multiply them, and creating the first droplet is a qualitative difference. Yeah? You can think of creating the first droplets as sort of the origin of life in some sense, yeah? And then having them it's just perpetuating it.
So,
Now in this case, you need fluctuations, and you probably also need chemical fluctuations. It depends on the system and the model, what you need to get it started. Yeah? So if you have a chemically active droplet, in principle you can nucleate it by some fluctuation, and then you need conditions where this happens with sufficient probability. But if you provide all the components, you will have also some D, because the reactions take place even if there's no droplet. But it depends on the rules that you provide. And I think there are versions of this where you may need chemical fluctuations, and there are versions of this where it's enough to have a fluctuation in a sense of classical nucleation theory to nucleate the first droplet. But it's an interesting question, and it's a complicated.
Yeah.
This one here.
So basically here for
very good.
simplicity, these only created within the boundaries of the droplet.
So I'm just discussing cases where I start as an initial condition with having a droplet, because it's simpler.
Yeah.
But there is a nucleation question. And I don't really answer it fully. I just give you hints about what it applies. Yeah.
Okay.
Yeah. Yes. So here I would like to show you a movie of a case where we only drive the system boundaries by reservoirs. Yeah? So this is a stable droplet. It has a fixed size because of the same arguments as before, except that now the non-equilibrium nature is only provided by boundaries. Yeah? And inside the box, detail balance is always obeyed, and this droplet undergoes an instability and divides. Yeah? So when we can sort of have models for dividing protocells that are just driven out of equilibrium by feeding them appropriately. And of course, if you stop feeding them, they disappear, they dissolve and disappear. At equilibrium, they don't exist. So, just to summarize this part, we think of this as a minimal model of a protocell. It's sort of a toy model which has many interesting features of the physics of droplets and chemical reactions, but it also has it can imitate many aspects of cells, but of course not in the biological sense, but in the physical sense. And it allows to discuss division, energetics, material supply, metabolism in physical systems. So it has a simple proto-metabolism, it has division and growth, it's far from equilibrium. So far, it depends on how you define far, because locally it's always equilibrium, as I explained. There can be different modes of energy supply. It doesn't need any membranes to function. You can, of course, try to add them. And so far, there is nothing that resembles genetics. So there's no evolution here. because there's no variability. But this could be added on top of this type of approach. Now, speaking of sort of the genetics, let's come back to the biology and to the biology of cells, and now think in terms of condensation phenomena in cells that can be used to organize biochemistry. I mentioned that this happens in the cell cytoplasm with these RNA protein granules, but it's also now an emerging theme that condensation seems to be very important to understand the organization of the nucleus and to understand chromatin organization. And I just highlight here two examples. So chromatin comes in different phases, which are called heterochromatin, euchromatin, more silent and more active chromatin. And this is also involves epigenetic modifications and modification of histones. And there's an emerging picture that condensation is a key concept to understand the formation of such domains and the organization of some domains. So the physical chemistry is very complex, but it's phenomenologically a condensation phenomenon, and it's out of equilibrium, and the concepts of condensates will be important here and are now sort of more and more studied. So this is now an interaction of proteins, histones in particular, and DNA that is organized in phases, in very complex phases. A second example is the regulation of gene expression. So the classical picture is that you have a promoter where your transcription factor binds, and then you recruit an RNA polymerase, and if everything goes well, it starts to copy an RNA from the DNA template and express the gene. Now, by labeling molecules in the cell and the nucleus, one often sees that this gene activity happens in localized structures, which appears puncta. And one idea is to think of transcriptional condensates as organizers of gene expression, where proteins condense near promoters, and maybe you have one condensate for several promoters that somehow then also can interact. And also enhancers may be part of that, and also join the condensate, which may explain why enhancer sequences can be at some distance from the promoter and still somehow regulate the activity of the expression of the promoter. So the picture is that what happens is a condensate which involves DNA and transcription factors that is key for the organization of gene expression. And with Jan Brugges and also with Stefan Grill in Dresden, we have been looking at very simpler systems to build up, sort of bottom up, these questions and ask, can transcription factors condense with or condense on DNA? And here, this is the experiment done in Jan Brugges' lab by attaching DNA at 2 points on a glass substrate. And then he adds transcription factors. And the starting point here are so-called pioneer transcription factors. There's some echo. An example is this molecule FOXA1. So these pioneer transcription factors are not specific to a particular gene expression. They somehow prepare the ground on the DNA. Just one thing here. Sorry. Thank you. Sort of prepare the ground on the DNA, but they are involved in regulation of transcription in a more general way, and they are therefore natural starting points to study such an interaction of molecules with DNA. And the observation here is that there is a co-condensation of this transcription factor with the DNA strand. So the attached molecule is here, and then there is a droplet forming, which pulls in DNA in the droplet. And which then also exerts a force on the DNA outside. And so here you see an example of such an experiment where the DNA is quite stretched. These higher intensity points are where the DNA is condensed, so that's a DNA signal, and this is the FOXA1 signal. And you see that where the DNA is condensed in this puncta, FOXA1 has condensed. And the picture is that we have such a co-condensation. And this can be discussed with very basic arguments. So I'm distinguishing here. I have the full contour length of the DNA, which is now separated in the contour length outside the droplet and the length that is inside the condensate, LD. And one can use a very simple droplet picture to understand these experiments. So first we can write down the droplet free energy, which now depends on how much DNA is in the droplet. And the first assumption is, and this can be tested experimentally, is that the condensate of protein plus DNA has a constant composition as it grows, which means that the volume is proportional to the length of DNA inside. And there's some coefficient alpha, which is in fact an area, which is the area, if you make a cross-section, the area per DNA molecule in a material. And with this picture, one can now write a free energy as a condensation free energy with proportion to volume. And nu is some coefficient describing the condensation energy per volume. And a surface tension term, which is proportional to area. And since the volume is proportional to L, then the area is proportional to L to the 2/3. And to this we have to add a free energy stemming from the outside strand, and then we can minimize the total free energy. Another way to think about the problem is to say there's a chemical equilibrium at the interface between DNA being inside and outside. So the derivative of F with respect to LD is not a force quantity, but it's rather a chemical potential-like quantity. And this chemical potential has to be equilibrated with a corresponding chemical potential of the strand. And then there's also, I'm just wondering, I may use a bit more blackboard. Do we have something to erase this? Or I'm not sure. There's also not so much time left. Maybe use a little bit this one here. Pardon? Yeah, that's still a bit. Just take a bit of this. I can explain this better in a moment. So for the droplet, I have a free energy, which is a function of LD. There I can define a new droplet, which is dF dLd. And for the strand outside the droplet, I can define, let's call this F polymer, which is a function of so that the stretched length, I'll ignore the size of the droplet for the moment, the stretched length and the length of the contour length of the polymer inside. And then we have the force that's acting on the ends is something like minus dF dL. And then there's a mu, which is more like a chemical potential, is dF dLp. And then the equilibrium condition is that these mu's are the same. And that's the balance condition. That's the wrong thing. Yes. Now, in order to discuss this second free energy of the polymer, one can start, this is sort of a more complicated calculation to do, but just to make it simple, we're using an approximation for the force extension curve. That's a famous approximation by Marco and Sitjar, where we say the force acting on the DNA can be written in a form which has the correct limits for strong stretching and weak stretching, and interpolates between them. And for weak stretching, essentially an entropic force, kBt over persistence length is here, and it will be proportional to the Hookean type of behavior. And then for strong stretching, it diverges when the contour length becomes the same as the extension. That gives this divergence here. And this equation somehow interpolates correctly between two limits and works very well experimentally. And now, in order to get the free energy, we can integrate this force over length. So the idea is we add to the free energy of the droplet the free energy of the stretched strand outside, and this is calculated by integrating the force over length. And this integral is easy for the equation I just gave you. And it gives me this term. This is now, this second term here is now this function f, which is a function of L and LP. And LP can be represented as LC minus LD. LC is the overall contour length. This experiment is done with lambda DNA, and LD is the size of the droplet. And now I have overall a free energy which depends on the extension that is imposed by the experimentalist and the size of the droplet, which the system has to choose. And the simplest picture would be to argue we now just minimize this free energy by finding the LD for which it is minimal and looking for the condition dF dLD is 0. And that's equivalent to saying these two chemical potentials are the same. And then by knowing LD from this equilibrium condition, we then find also what is the force from the force equation. Now, because the system is very small and fluctuating thermally, there are fluctuations of LD. And so what one can do is one can plot the free energy as a function of LD. So we keep L fixed because in the experiment the endpoints are fixed. And if we now vary LD, we change the free energy, and this would be the potentials that you get. And L here is the endpoint distance. So this is for endpoints. If the endpoints are very near each other, then you get large droplets. The minimum is a large LD, and if you now stretch the endpoints, then the droplets get smaller. The minima move to small LD. And using Boltzmann factors, these potentials can be turned into the distributions of LD that you would expect in an experiment. And now this fluctuating model captures the experiments and can allow to measure these parameters that I have in the simple model, the surface tension and the condensation energy. Here you see the LD is a function of L. So the droplet sizes are large when L is small. These are data points in the simple model. And this is sort of a first order transition between having a droplet and not having a droplet, depending on length. For the axis is moved out of the image here. This would be the probability to have a condensate as a function of extension. And there's a jump-like change to the first order transition. Okay, so I think I have about 5 minutes left or so, but I will probably come to the end now. Maybe just finishing in the group of Stefan Grill, a similar experiment was done with more stretched DNA, so that it was not able to co-condensate with the transcription factor. So here the stretching is stronger between two optical tweezers. And now what one looks at is how small condensates form on the DNA without the DNA being also condensed. And in this case, in this experiment, the concentrations are also below the concentration where the proteins would form droplets in the bulk. So the system is not yet in a phase separating regime, but it forms small condensates on the DNA. And you see here a function of time how these condensates form along the lambda sequence. And here the picture that arises is that we see something like a pre-wetting phenomenon, in the sense that there is a ability to form condensates below the saturation condensation, where it would form macroscopic droplets in the bulk, because it is sort of helped by the surface interactions to bring molecules in proximity. So in general, if you have a phase coexistence, a phase diagram, and you bring wetting into the picture, then you can have a transition between complete and partial wetting if you have a real phase coexistence. But outside of the phase coexistence region, you can have a transition between a first order transition between a thin layer and a thicker layer below phase separation. And that's called a pre-wetting transition. And we think we see such a pre-wetting transition in the system, and there's a sort of the signature is that the intensity makes a switch between a very thinly adsorbed states where just molecules more or less bind individually in the surface, and the formation of a real condensate, but which is a condensate that is small in size and cannot really grow because the system is not supersaturated. And what is quite exciting in the system, and I will just sort of, before I finish, just allude to it a little bit, is that we can understand here the effect of sequence of this pre-wetting condensation phenomenon. So this is done with a transcription factor called KLF4, and it is known that it has certain binding motifs on DNA, and they are usually represented such logos where you say which bases should be at certain positions in order to have a high probability of binding. And one can translate now this logo, given a sequence of DNA, like the lambda sequence, into a distribution of probabilities where you would expect to find a molecule. And this looks like this here. And we take this as an input for a very simple condensation model, which is essentially an easing model, where we say we have a condensed state, well, this also breaks down, condensed state and an absorbed state, and they correspond to a two-state system. And there is a field which favors one or the other state, and this field comes from the overall concentration, which is homogeneous, but plus a modulation by the DNA sequence, which we can infer from this binding motif. And on the right-hand side, you see a comparison of such a model. So the upper image is the distribution of condensates in many experiments on this lambda DNA, and it gives a pattern which comes from the sequence. And the lower image is a realization of many of the statistics of an easy model with a kind of, we wouldn't call it a random field, but with a field that is modulated by sequence as taken from the DNA. So we have here a condensation phenomenon that is helped by the binding to the DNA and is organized with the help of the sequence, but not in a way that single molecule interactions determine the binding, and because the condensation is a collective phenomenon, so it's more patterns of the sequence on larger scales which govern the physical chemistry of this condensation. So this gives a slightly different picture of how to think of DNA-protein interactions when having condensates, and of course they may be relevant in the case of these transcriptional condensates. Okay, so I think the time is up. Thanks that will allow me to speak longer as we started late. But I should stop here, and thank you all for your attention.
Thank you. So thank you for the very nice talk. I have a general question about the question you raised. On the one hand, you have transport phenomena. On the other hand, you have chemistry, and you want to study the relationship between the two. I must say, I'm really surprised you don't say a word about Peter Brian Mitchell theory of chemiosmotic.
About which I didn't understand.
About Peter Brian Mitchell and chemiosmotic theory. I mean, it seems to me, I may be wrong, that you treat chemistry as a scalar phenomenon. I don't know where the transport comes from. That's my general question about this.
What comes from?
Where does the transport come from in your system? you have this sentence which is very clear. We don't care where ATP comes from. Well, in fact, if you look to an ATP synthase, you do care.
Yes.
So that's my question.
Okay.
How do you compare this with the coupling theory of Mitchell?
Yeah. So the general framework which I described, of course, allows to integrate all these elements. Yeah? And also the models I outlined, they can be used to introduce metabolic elements. It is just that sometimes it's useful to not include it in a model. You know? So when I, for example, take the model with the supply with reservoirs from outside, that's sort of in the spirit where everything I take into account, I know where it comes from, in some sense. Yeah? So I explicitly provide nutrients and waste. And then I can have versions of the model where I don't explicitly provide them, but just assume that they're there. And these are just two different ways which are suitable for certain conditions. For certain questions, it's better to use one type of model, and for certain questions, it's better to use the other type of model. But the general formalism allows to take everything into account if you need to. You know? So this is a very general, versatile framework. And then depending on the question that you're asking, you can simplify in different ways. That's the way I think about it.